In the first part of this presentation, we're discussing complex data structures and kind of statistical approaches to incorporate these complex data structures in the analysis. And I talked mostly about data, complex data that would arise from the same individuals, maybe observed a few times. But what about data on tracts and streamlines? How can we deal with multiple tracts within the same individual? What about the data on the streamlines within tracts? Well, fortunately, linear mixed models can deal with most aspects of such data. And this will be more in a framework of hierarchical data and hierarchical data analysis. Now, one topic that we'll not address today, it will address hopefully in the next year's DIPY workshop, is how to properly model spatial data along the tracts, along the streamlines that we do know that data are highly correlated between, let's say, when they lie in close proximity on the streamlines. We'll be using a tool called functional data analysis, but again, this is not for today. This is for the next workshop. So, streamline data. You have already seen how this data are generated in DIPY. So here I won't go deeply into the extraction of the data because this has been covered in other presentations. But let me just go briefly over that. So what we do, extract the bundle, we estimate the bundle centroids. We then divide those centroids into a fixed number of so-called disks. For example, we can divide a centroid into 20, 50, 100 disks. Now, the important point here is assignment of each point on the streamline to the closest centroid disk. So this is done within DIPY. And then what we can do, we can extract data. Let's say we can extract fractional anisotropy for, let's say, bundle B, subject I at disk D. And this is how we'll denote such data. It's FA for subject I in the subscript, bundle B superscript along disk D. So we published the whole pipeline of the analysis. I'm describing today the statistical analysis that was done in this paper. This paper appeared not so long ago in the scientific reports. Paper is called Bundle Analytics, a computational framework for investigating the shapes and profiles of brain pathways across populations. So, as I was saying, first the bundle is extracted, then the model bundle centroids are extracted from the bundle. And then you can see here in different colors, these are different disks along the centroid. So the important point here is this assignment map in point D, where for each disk, we assign all the points on the streamlines as belonging to that disk. So this will be our units of analysis that we will analyze. So we might extract FA values on such a bundle. And here is just a presentation, you can call it a heat map of the values, where we see the large values here are in kind of yellowish and orange color. And we can see them along or within this bundle at some points. Majority of the points here of the values, 0.4, 0.6, and at the ends, both ends of the bundle, those values drop to like 0.2. So now, if we actually extract such a data within one of the bundles for one subject, and here I'm just showing data for 200 streamlines. Some of the things you might notice is that data on a streamline might be pretty noisy, but the nice thing here is that this data are registered or aligned along the disks. So you can see here on the x-axis, we have data for 100 disks. And as I said, we have observations here from 200 streamlines. Now the question is how to incorporate all this data in the same kind of analytical model. So here, for example, one of the things that we can do also is we can extract the data only from the streamlines that are, let's say, closest to the centroid. And such an option is already implemented in the pipeline. So let's say you might want to say, well, I only want to extract data from the streamlines that are, say, 2 millimeters from the centroid in this bundle. So a simple summary of such data might be, well, at each disk, we'll calculate the mean and let's say standard deviation for one subject. So we could use such data for the analysis, but I didn't introduce these linear mixed models earlier to just stop at this point to use just the summary data. So what data do we have? Well, we have actually data here for streamline S, for subject I, bundle B, at disk D. So what we'll do, we'll treat all of those points as data points, and we will model the whole data for all the streamlines, all the bundles, for all the subjects at all the disks using a model as I'm presenting here. So we might have this beta 0, which is a population mean for track B. And then for each subject, we might have this UIB, the random effects for subject I, bundle B. And then data that we collect on streamlines, of course, will be away from both the population mean at disk D, but also from the subject mean at disk D, because we still have hundreds or thousands of streamlines that we're analyzing. Now you might ask, well, this was nice for one group, but usually we want to compare two groups. Well, that's not difficult because we're just introducing another fixed effect here, beta diff, which will correspond to the mean difference between two groups at disk D. In the next and last part of my presentation, I'll show some results from this analysis. And these are the results that we summarized in our paper, in the BUAN paper.